Good morning, everyone. Sorry we're a few minutes late, but I'm Lorraine Maxwell, and our guest for this morning has signed in. So I've run Zoom meetings, but I haven't run a webinar. And I think what happens with webinars is that participants have to type in their questions in the chat or the Q&A. So we'll hear from our guest first. I will introduce him in a minute, and then we'll have time at the end for your questions and comments. So our speaker this morning, our guest this morning is Dr. Hector Aguilar-Carreno. He's an associate professor in the Department of Microbiology and Immunology at Cornell, here at Cornell. He's been with us since July of 2017. He comes to us he did his bachelor's in Mexico and then immigrated to the United States and got his master's from California State in Los Angeles and his PhD in biochemistry and molecular biology from the University of Southern California. He did his postdoctoral training in virology at the University of California, Los Angeles. He is known for adopting technologies previously foreign to the field of virology to the study of enveloped viruses, which he will explain to us. And he also studies viral entry and assembly-informed novel ways to develop antivirals and vaccines, which I'm sure we're all very interested to learn more about. Here at Cornell, he's been, since his time at Cornell, he's been an ad hoc member of NIH study sections, a standing chair and member of the American Society of Microbiology Committee for Minority Education. He has chaired Cornell's College of Veterinary Medicine Diversity Committee and served on a number of PhD committees and has chaired 7 of those students. I also have to say that he and I know each other in another way. We both sang in the choir. Well, we will, again, hopefully soon, sing in the choir at First Presbyterian Church. And so I am very happy to welcome Dr. Aguilar-Carreno this morning. Thank you.
Thank you so much, Lorraine, for your nice introduction. So I can't see everyone, so it makes it a little harder to give a talk, you know, when you can't see reactions in people, but I'll do my best to hopefully explain things the right way for everybody. I imagine that all the participants are from very diverse backgrounds, but all very smart people. So I'm gonna push a little bit your comfort zone maybe and try to explain some concepts that might be a little bit difficult, but hopefully enjoyable to learn about. And so I will share my screen now and hopefully everything will go smoothly. Okay. Can everybody see? Maybe I can get a head nod from Lorraine who I can see. Can you see my screen, Lorraine? Okay.
Yes, we can see it, yes.
Awesome, great. So I'm gonna get started here. So the title of my talk is Emerging Deadly Viruses and Their Glycoproteins from Infection to Vaccines. And so what I'm gonna do is, let's see, okay. All right, so I'm gonna start by telling you a little bit about a few interesting facts about envelope viruses. See this list of viruses here, the virus family on the left, all the virus families, the majority of them are envelope viruses. These are virus families that have been found in bats in the last decade or so. And notice at the very top of the list is the Coronaviridae family with about almost 4,000 species of these viruses found in bats. followed by rhabdoviridae, so that includes rabies, VSV, and then the paramyxoviridae includes Nipah and Hendra viruses. So these are viruses that you may have heard of. So obviously you've heard now of SARS-CoV-2, which is the virus that is causing the current pandemic, COVID-19. Just for terminology purposes, so the virus is called SARS-CoV-2. The pandemic is COVID-19, right? So the virus is not COVID-19, and it's COVID-19 because it's coronavirus disease, and it started in the year 2019. So SARS caused an epidemic, a big outbreak a few years back, then MERS, another outbreak. And so these are, again, enveloviruses that you might have heard of. Another important one is Ebola virus that you probably heard of that caused an outbreak in 2014. And so the interesting thing is that there's a huge predominant of envelope RNA viruses in these viruses that are more likely to cause pandemics or zoonotic events, so jumping from animals to humans, basically. And a lot of them originate in bats. So it is a very interesting group of viruses. So the next slide I'm going to show you is some of the viruses that we work on in my lab here at Cornell. And so we have a big list of viruses that we're currently working on, and I'm going to touch a little bit on a few of these that we're working on vaccine development for, which I thought you might be most interested in. So Nipah virus is one of the main ones that I've been studied since I started my lab and since I was a postdoc at UCLA. So basically since 2003, I've been studying this virus, so almost 20 years now, and 18 years or so. And then what I want to say is that these viruses come from fruit bats. There's two main genera, Pteropus and Eidolon fruit bats. Very deadly viruses in humans. They can jump from bats to pigs, and then pigs to humans, or directly back to human, and also human to human. And they can reach 40 to 100% mortality in humans. So really, really devastating, deadly viruses. They have a wide mammalian host range, so they can infect, from bats to, pigs, horses, dogs, cats. And so they have a lot of potential for causing one of the next pandemics. So that's why we're so interested in studying them and coming up with vaccines and so on. So there's a wide distribution of these bats in Asia, Africa, and Australia, and a lot of them are seropositive for these viruses, which means that they have seen the viruses, they have carried them in their bodies at some point. And so because of all these conditions, all these modes of transmission, these are very important viruses to study. My lab has been focused, and don't worry, you don't have to understand this big structure here, but I just to illustrate that my lab has focused on the viral glycoproteins on the surface of the virus, and that's what's in my title, right? Why do we focus on the viral glycoproteins? There's two glycoproteins on the surface of Nipah virus that's represented here with in this simple cartoon. They're the F and the G glycoproteins, okay? So these glycoproteins on the surface have two purposes. One is to bind the cell receptor. So you imagine a cell right here, that one of the glycoproteins can bind the receptor, and the other glycoprotein can fuse the viral membrane, a lipid bilayer on the surface of the virus, to the cell surface. And that allows viral entry. It basically allows the virus RNA that encodes the genetic material of the virus to get inside the cell. Once inside the cell, then the virus can replicate and make more virions. And so from one virus that infects one cell, you can have thousands of viruses coming out of that infected cell. So my lab has been studying the functions of the glycoproteins because of several reasons. One, they're very important for viral infection, as I just explained. But the other reason is that they're very important for vaccine development. Most of the vaccines, the antibodies that our bodies make against these viruses are actually made against these glycoproteins. They neutralize, we call it neutralization. They neutralize, they block the function of these glycoproteins. So they binding to the receptor. Here in this diagram, I'm showing the receptor for Nipah virus, which is on the cell surface. So now I'm switching it back. Here's the cell on this side. Imagine this line up here is the cell. So this is the receptor sticking out of the cell. And here's this G glycoprotein from the surface of the virus. And so there's a series of changes that these glycoproteins undergo that we've discovered in my lab that are important for effectivity. But also this same protein right here is very important for neutralizing. You can have antibodies. Your body can make antibodies that can block one or the other proteins. And that's how you survive a viral infection, okay? The problem is that, you know, I told you this virus has 40 to 70%, so 100% mortality in humans, average around 70%. And the problem is that our body cannot make the antibodies quickly enough to fight the virus. And so most people end up succumbing to the infection. And so vaccines help so that your body can learn how to make an antibody against a specific virus, and then later on when you get the actual virus, you'll be protected because those antibodies will be made really quickly and will counter the viral infection. Okay, so I just wanted to say that we've been studying these glycoproteins for, I've been studying viral glycoproteins for the last 25 years since I was a grad student. And so that's a reason to then use these glycoproteins and what we know about them for vaccine development. I want to tell you about 3 vaccination strategies that we do. We have 3 different platforms for vaccine development in the lab. One is called viral-like particles, where you take basically, these are the glycoproteins I was telling you on the outside of the virus. And these viral-like particles is basically the outside, okay, of the virus. So it is the shell, so to speak, has the lipid bilayer, the glycoprotein, some internal protein. And you can put, actually the beauty about these particles is you can put multiple glycoproteins from different viruses onto the same particles. And then we thought about this idea in recent years, in the last 3 or 4 years, and you can actually protect maybe against multiple viruses at the same time. Because if you notice, these particles don't even have any genetic material. They're not they are not replication competent. These viruses cannot infect cells, but they cannot be replicated in cells. And so they can only enter once. And so they're very safe. And still they can produce immune responses that allow antibodies to be made to protect you against those viruses. The other very similar type of vaccine platform is called pseudotype variants, pseudotype viruses. And here is an electron microscopy image of one of those variants. And you can kind of see at the surface here, hopefully, a little crown of glycoproteins on the surface. So these are basically what this cartoon is representing, is this real virus. So there's some internal proteins inside, and this is the difference between these two is dangerous to humans. It doesn't replicate, it's only entering the cells once, similar to this, but sometimes it can incorporate viral glycoproteins such as these very effectively, and multiple of them. And so another way to make multivalent vaccines. Another way of making vaccines is by inactivating the actual virus. So for example, the vaccine that that people are receiving right now for COVID-19, for SARS-CoV-2 in China and in Russia, is this type of vaccine where you have the virus itself, and you inactivate it chemically or by temperature or by some means, and then you can use the dead virus, right? The virus that you have inactivated as a vaccine itself. So it has all the components there. It doesn't always work very effectively. So we actually did some of the experiments with a dead SARS-CoV-2 virus that I'll tell you about later. And we actually, we got about 60–70% efficacy, but it's not as good as some of the other vaccines out there. Okay, so I'm going to focus most of my talk on these vaccines, and I'll tell you some exciting data that we have against these viruses. So the first one I'm going to tell you about is sorry, I'm going back a little bit. Is this a pseudotype virus vaccine? This type that a multivalent vaccine that has worked really well to protect against Nipah, Hendra, and Ebola viruses. So the first thing I'm going to show you is this map to show you that why we're doing this vaccine. Why do we want a multivalent vaccine against these 3 different viruses? Well, the reason is because these viruses all come from fruit bats, okay? And then the habitats of those fruit bats actually overlap. So here's, for example, the habitat of the Pteropus fruit bats that can be found in Southeast Asia and Australia. And this is where most of the Nipah virus outbreaks have been observed since 1999, when this virus was first identified, up until last year. And basically yearly outbreaks of this virus happen. Most of the outbreaks have concentrated for Nipah virus in Bangladesh, India, Malaysia, Singapore, and then a very closely related virus, Hendra virus. There's been multiple outbreaks in Australia. And now these viruses have also been found in all these areas right here, such as Ghana, Cameroon, and I bet you anything, if we keep looking, we'll find them in bats in other countries in Africa. Ebola virus, as you probably remember, in 2014, that was an outbreak that flourished mostly in Africa, and so all these pink colored countries are where the outbreak mostly happened. And you see seropositive fruit bats in all of these countries frequently. And so there's a lot of overlap, as you can see in this map. So that gives us justification of making a multivalent vaccine that would protect against all of these viruses simultaneously. And that vaccine could potentially be given to people in those regions, to the US Army when it goes to those regions, to animals, to livestock, so that the virus will not jump from, for example, from bats to pigs to humans, so that pigs could be vaccinated and so on. Okay. So here is just to show that we have optimized viral-like particle production to include different proteins from these 3 viruses, Nipah, Hendra, and Ebola virus. This is some Western blot showing that. Other ways to look and add the viral particles. This is basically what the vaccine looks like, the virions. So these are viral-like particles. And this is the same thing, just to show you that the picture that I showed you before, where we can put multiple glycoproteins in the same virions. Okay. One thing that you have to think about when you make these vaccines is thermostability. Why? Because most of those countries that I just show you in the map are have tropical weather. So they're going to be very hot temperatures most of the year. And so you don't want to make a vaccine, right? And you're going to administer it in those countries particularly. And again, we are doing it there to protect us here, to protect them, but also to protect us, right? Because as you know, from this pandemic we're living in now, once you have a pandemic, it can spread all over the world. And so you gotta go to where it's potentially going to start. Anyway, to vaccinate people in those countries and in many places in the world, in Latin America and in many places, you will need something that is thermostable. Even here in the US, the vaccines that we are receiving right now are not very thermostable. The RNA vaccines, and I'll tell you a little bit more about it, but they need to be kept in the cold. And so that makes distribution and application very difficult, especially in developing countries. So here's an example of a vaccine that we took, the VSV vaccine that I told you about. If you focus on this black line, this is how long the vaccine can be good. Basically, the virus can be intact and enter cells well, and at 37 degrees, this black line right here. And so you can see that at seven days, just storing the vaccine dry, basically in a vial, you lose a lot of activity. By the time you get to 14 days, you've lost all of the activity. This is the negative control, basically, where you have no activity at all. So you start with really good activity. This is a log scale, so you lose like 3 logs of activity. 1,000 fold activity, basically. And so this is a very fast degradation. With lyophilization, we found a system to lyophilize, dry up the virus in trehalose, a type of sugar, and we can extend that life for about what is it, 35 days, 5 weeks, right? And at 37 degrees. So this is a very high temperatures. This is 37 Celsius, so it's about close to 100 degrees, 98, 99 degrees Fahrenheit. And so we made our vaccines a lot more stable. Then what the next thing we did is we took these vaccines and then we put them into hamsters. And I'm going to show you a lot of, I hope you guys don't have a hard time understanding why we do this, but I'm happy to explain that at the end, why we have to use rodents, mice, or hamsters to do the testing. Obviously, we're not going to put this vaccine into a human, right, without having first tested it in a small animal. So in this case, we use hamsters because they're a good model of disease for Nipah, Hendra, and Ebola virus. So we loaded this vaccine with the glycoproteins from all of these viruses, as you can see here, and then we put them into hamsters, and then we vaccinated twice, and then we got serum from those hamsters. So we collected blood, and we looked at the blood, and what this is showing is the blood from the hamsters, the antibodies in the hamsters were vaccinated with a VLP vaccine, were able to neutralize Nipah and Hendra virus really well, and Ebola virus so-so. The hamsters vaccinated with the VSV vaccine were able to neutralize the infectivity of all these 3 viruses really, really well. And so we decided to go on with the VSV vaccine and actually challenge these animals. So we vaccinated a group of 36 animals. 6 animals per group were challenged with different viruses. So 6 vaccinated, 6 unvaccinated, challenged with Nipah virus. 6 vaccinated, 6 unvaccinated with Hendra, and then the other 12 with Ebola. And this is what we did. So this is a vaccination period. This is how we organize this vaccination in time. And so what we see is that we challenged then the animals with Nipah, Hendra, or Ebola virus, collected blood, and did clinical studies, pathological necropsies, and all that to see how the animals behaved. And the beautiful, beautiful, amazing news is basically, I want to focus your attention right here. This is temperature, clinical scores, which we don't have to go into detail. This is weight loss of the animals. So the beauty is right here, survival. 100% of the animals vaccinated with this multivalent vaccine survived either challenge with infection with Nipah virus, Hendra virus, and Ebola virus. 100% of the non-vaccinated animals we had to euthanize in days 5 through 9 when the animals are getting really sick or they lost more than 20% of their weight. Basically, we humanely euthanize the animals so that they don't continue to suffer. So that's the criteria that you use, because those animals are going to die if you continue to leave them alive. And so we had to euthanize them during days 5 to 9 of the 16 or 15-day trial. And for again, all of our animals survived. So this is really, really encouraging. We got 100% efficacy with this vaccine against those 3 viruses and 100% safety. None of the vaccinated animals had any issues whatsoever with the vaccine that we could detect clinically or in any other way or pathologically once we did necropsy on the animals. So this is a study we're right now about to submit for publication. Here's just to show you some of the tissues, some of the pathology that came out of those animals. And so we had, this is Nipah virus, an animal vaccinated, challenged with Nipah virus. This is the brain and this is the lung. And so you can see a lot of inflammation around the blood vessels here in the brain of mock vaccinated animals, a control animal. But the multivalent, the vaccinated animal with our vaccine, the blood vessels look very different, right? From here to here, very normal and not inflamed. So this is a sign of encephalitis, basically here. So in the lung, lots of inflammation. See all these red cells? There is like lots of inflammation coming into the lung and just clogging the lung. The lung, this is what a normal lung looks like in the vaccinated animal. Lots of space. That's where you get the oxygen. That's where you're breathing. Here, lots of clutter. So you can't. This animal was having a hard time breathing, and so it's one of the animals that was euthanized. For Ebola virus, it affects the liver a lot. So you can see, again, a lot of that red color, a lot of immune cells coming into to the liver to try to fight the disease. And this is the vaccinated animals, which is what a healthy liver looks like. Same thing here with the lung, lots of space here, some inflammation in the lung also for Ebola virus, less than for Nipah, but a good amount of inflammation. One thing that came out that is really nice also that we discovered with this vaccine is that not only the neutralizing antibodies were made against Nipah, Hendra, and Ebola virus, as I showed you before, actually they were made against a related virus, which was incredible. It's called Cedar virus, is in the same family as Nipah and Hendra, and no vaccine ever made against Nipah or Hendra had been able to neutralize a different virus. So we think that was, so here's the neutralization. This is antibody binding to Nipah and glycoproteins, and this is antibody binding to the F protein of a Cedar virus, and then neutralization of Nipah virus here 100%, and of Cedar virus about 60% or so neutralization. So this is really, really exciting because it says that by putting multiple glycoproteins in the same virions, in the same vaccine, in the same viral particle, you're actually prompting your immune system to make antibodies that are cross-neutralizing, we call them. So they're binding to conserve epitopes, conserve parts of the proteins of these glycoproteins, and they're neutralizing beyond even the viruses that you vaccinated for. To other related viruses. So it's a very, very interesting finding that it's a bonus of our study. And so we're pursuing that further. Okay, so the question is now, can we apply this vaccine platform and this knowledge base towards a pan-coronavirus vaccine? And what do I mean by a pan-coronavirus vaccine? We know that the vaccine that we are hopefully all about to get, or going to get soon for SARS-CoV-2, for COVID-19, targets basically one virus, which is SARS-CoV-2. What we're interested in my lab right now is making a vaccine that would protect you against all of the related viruses, so that no matter what strain or even other coronaviruses, SARS-CoV-1, MERS, all the related viruses, you'll be protected against them. And so because of the really beautiful results that we got with the Nipah, Hendra, and Ebola vaccine, we believe that we can use the same platform to protect, to have a coronavirus vaccine. And because and I'll tell you what, the S protein, the glycoprotein from coronaviruses serve the very same purpose, and actually even have very similar structures to the Nipah, Hendra, and Ebola vaccines. So here's a little bit of that knowledge here. So here's a coronavirus cartoon now, and this is the protein I was telling you about. The coronaviruses actually have 3 glycoproteins, S, M, and E. And they have different functions, but the one that is the main one for eliciting neutralizing antibodies is the S protein, and is the one that has the same functions as the ones I told you before, the F and G and GP glycoproteins for Nipah, Hendra, and Ebola virus. And so we're making viral particles with this protein, but we can also incorporate these two, if necessary, these other proteins into the virions, just as this picture shows. And that's what we're working on now. I want to tell you a little bit about the types of vaccines that are being out there for coronaviruses. There's, you might have heard all this terminology lately, the recombinant vaccines that has the S protein. All of them are based basically on the S protein, except for the inactivated virus vaccine that I told you, this is a dead vaccine. So RNA vaccines are becoming more and more popular, and that's what the Pfizer and Moderna vaccines are. And then there's live attenuated and the pseudotype vaccines, replication incompetent vaccines. So here's the virus, the spike proteins. And so all of these are different types of vaccines that people are trying to generate and around the world, in companies and different countries. So what I just wanted to show you this table showing all of the vaccines, or not even all of them, but some of the vaccines that have been tried, okay, and what made it into phase 1, phase 2, and phase 3 clinical trials in humans is, a handful of those. And these are basically the licensed vaccines. At the time when this was done, it was just this replication competent vaccines, inactivated vaccines, and a couple of, here's the RNA vaccines that we're actually now licensed as well. So we can move these blue ones onto this license column right here. And so all of this basically, again, are based on this spike protein on the surface of the virus. And so we are trying to put that protein then in our multivalent platform into these particles. And so this is just to show you that we have optimized how to do that, because it's not that easy. We tried many different types of ways of putting the S protein here. And this is showing viral entry of those particles, which is a way to measure how good they are. So this is our negative control, and all of these are different levels of entry of the virus. So the higher, the better. Basically, we're looking for something high. So the optimized systems that we have here have basically, this is a NEPA system. So this is our control for our goal, what we had before, what I showed you before, those particles in the vaccine. And this is the system, how far we've gotten so far to get to basically the same as signals as the NEPA vaccine. And so we are very happy with these results. And now we are trying to do the multivalent incorporation of the multiple S proteins from different coronaviruses, and trying to get funding right now from DARPA and from NIH to attempt this. So the next part that I'm going to show you is basically what we have set up already in the animal side of things to be able to test our coronavirus vaccines. Because once we have the vaccine ready, we obviously have to test it in animals. We'll want to test it immediately. And so we've done 3 types of animal models. One is we used a mouse, or kind of a natural coronavirus in a mouse model that infects mice, and it's called mouse hepatitis virus. And so you can guess from hepatitis that infects the liver. We also use a transgenic mouse model where the receptor for the virus is called ACE2, human receptor, has been put in the place basically of the mouse receptor. The wild type mice cannot get infected with SARS-CoV-2, but then if you put a human receptor in there, then you're making the mouse susceptible to infecting with the actual SARS-CoV-2 virus. And the other model is a hamster model. Hamsters can be just naturally infected with SARS-CoV-2. Their receptor, their ACE2 receptor, is more similar to human. So we've worked really hard. This last year has been tremendously hard with COVID, and having a lot of homeschooling at home, but at the same time trying to get help the pandemic as much as we can in vaccine development and antiviral development. So thanks to a lot of people here at Cornell, the IBC and BSL-3 staff, animal care staff, we've gotten these models set up. All these 3 models are set up now, and we're ready to test the vaccines. So we've done actually a little bit of testing already of one of the vaccines, not the multivalent vaccine, but another one. Just want to show you in this model, the ACE2 mouse model, where we put in the human receptor. It's called a K18 model, human receptor into the mice. We infect them with different concentrations of the virus. And you can see, the more virus we put, the earlier the mice succumb to the infection, and we have to euthanize them. So all of these are pre-studies that we do before we can study the vaccine efficacy. So here's just showing some tissues from those animals, and that's showing the inflammation that I showed you before, basically, in the lungs of those animals, and in the brain as well. So we have good models now that we can use for testing of our vaccines, and also therapeutic approaches, drugs basically, that may help the infection. This is just, I'm gonna jump through this. This is some just showing that we can look at immunology, also B cells and T cells, immune cells from those animals, and study those responses as well. Okay, so far, basically, we have, this is, I need to update this slide, I'm sorry. It says, we'll finish in 1 week. We just finished the hamster model last week, and we're about to start testing of some of these multivalent vaccines, hopefully very soon. And so really quick, and I think I may skip some slide. I want to make sure there's time for questions. And we have, Lorraine, we have until 11:30 total, right?
Yeah, 11:30, 11:45. Yeah.
Okay, all right. So I'll try to end a little before 11:30, so we can have some time for.
Yeah, we already have a couple of questions. I don't know if you want to take those now or later, Hector.
Maybe we'll do it at the end, if that's okay. Yeah.
All right.
All right. So I'll try to finish. I'll skip some slides so we can finish in time. So just wanted to tell you a little bit of this new method of inactivating particles, making a new type of, a different type of vaccination. This is based on a chemical that we put on the membrane of the virus. And with that approach, we actually have shown that this method can inhibit or kill basically a lot of viruses, every single membrane virus, right? Because you need the membrane for the chemical to act on, can be killed with this method. And so here's an example of influenza virus infections and how you can reduce it by adding increasing amounts of this compound. So the beauty about this compound is that it intercalates into membranes, it reacts with lipids, it inhibits membrane fusion and viral entry, but it leaves the glycoproteins in those virions intact in their native conformations. So as much more than the vaccines that we're getting right now, for example, for flu, the flu shot, the influenza vaccines, those are inactivated viruses, but they're inactivated in a way that they don't preserve the conformation of the glycoproteins as well as this new method that we have. So we are hoping that we can improve vaccine development for some things like influenza and perhaps obviously other viruses, coronaviruses included. And so this is ideal for development of envelope viruses. And I'm just going to show you some exciting data where we tested this. This is the chemical I'm talking about, this purple line here, XMO1, okay? So follow that versus the traditional, where I have traditional, and it means the traditional influenza vaccine that we get, a flu shot that we get every year. So out of 10 animals, you can say here the percent survival. For the influenza, for the traditional vaccine, 80% of the animals survive, okay? Which is good, it's not bad, right? For the XMO1 inactivated virus, 100% of the animals survive. When you look at the weight loss, okay, the the traditional vaccine, you see this much weight loss. They went to about a little below 90% in their body. So they lost 10% of their weight. So just to give you an idea, if you lose 10% of your weight, that's a lot of weight, right? If you weigh 200 pounds, that's 20 pounds that you lost. And this is obviously over a period of 2 weeks. So that means a lot of disease. They're having a lot of clinical symptoms. Our vaccinated animals that were then challenged with influenza, they lost almost no weight, this purple line, okay? And so that is really, really amazing that these animals are doing so well. This is the antibodies made against one of the glycoproteins, the neutralizing antibodies made against one of the glycoproteins called HA or hemagglutinin. And you can see the traditional antibodies, well, they can protect basically 40% or neutralize about 40, it's called an antibody titer, 40 as compared to over 100 or about 100 for the XMO1 vaccine. So you're making a lot better neutralizing antibodies for HA and also for another glycoprotein, the NA. So this is the traditional, the brown line versus the purple line, which is, you're neutralizing these antibodies a lot better. So we use this approach. So this is very exciting, and we wanted to try the approach against the coronaviruses. So far, we haven't gotten this to work very well. We need to optimize still the way we inactivate those viruses with this compound. As you can see here, the animals are not very well protected. But the traditional virus inactivation, some of those vaccines that are being made, I believe in India, people are receiving the inactivated vaccine as well. They have some protection. So some of the animals are surviving, as can be seen in these graphs, with this virus, and as well with the SARS-CoV-2 virus in another vaccine. So here's the non-vaccinated animals. They all succumbed to the infection. Here's the XMO1 vaccinated animals. They all succumbed as well, unfortunately. But the PFA, the traditional method, right? This would be the equivalent of the traditional virus inactivation method. But half or a little more than half of the animals actually survived, which is good. It's just not as good as we hope. So we're still working on this. And here is just some data showing you the hamster model that we have set up, where we can put different amounts of concentrations of the virus and get different various results for weight loss. And so I'm gonna end. Okay, this is I'm just showing so far the viral loads in those hamsters. I'm gonna end with some very exciting results. This is my last data slides that I'm gonna show you, because again, I want to leave some time for questions. This is a chemical. It's called N0385 that we have tested recently, just in the last 2 weeks. This is literally data fresh off the oven, right? So we finished this experiment last week, late last week, and this is a chemical, okay, that we got from some collaborators in Canada that inhibits an enzyme in the cells that the virus, the SARS-CoV-2 virus, needs for viral entry, for infection, basically. And so if you're knock down this enzyme in the cells, which is basically not required for survival, right, of the cell, you're actually knocking down infection with the virus. So they have done this already in cells. They in vitro, it's called, right? And they have seen some good activity. And they ask us, since you guys have the animal system set up, can you guys test that in your animal system? So we chose the animals, the mice that have the human receptor in them. And what we did is we infected the mice with the virus, and then we kind of follow them over 14 days. As you can see, this is the saline, the control, basically, where we're just putting, water, basically, with physiological salt levels there. So no chemical at all, no drug. All of the animals, 10 animals, 5 males and 5 females, all of this succumb to the infection, as we knew they would, right? From days 6 through 8. So between them, that we have to euthanize all 10 animals. These are the animals that received the drug, and look at this: 7 out of the 10 animals survive. All the females survive. And in this model, it's very much like humans. Males do worse than females when they're infected with SARS-CoV-2. More males die than females, as it is, again, the case in humans. But even 2 of the males survived, 2 out of the 5, and all 5 females survived. So this is really encouraging. And guess how we administered this drug? We actually did it intranasally. So just put 1 daily dose, 1 drop of the drug on each nostril of the mouse. So intranasal, okay, just put a drop in there for a couple minutes, and then that's it. 1 treatment a day, one dose a day. So this is amazing results that we have, that this drug can actually protect the animals, and we only gave the drug from 1 day before we infected the animals through day 6, and then we stopped. And even then, a lot of the animals went on and survived. So we stopped. I think if we had given the drug all 14 days, we might have had even more survivors. This is just a control with an inactive drug, but similar drug. And 1 animal, 1 female survived out of the 10 used in that control drug. You always have to use that as a control, as a scientist, to make sure that your active drug is doing the effect because of its activity. So here's my conclusion slide. I show you basically that multiple viral glycoproteins from multiple enveloviruses can be incorporated into single viral particles. And that vaccine can produce highly and broadly neutralizing antibodies against these 3 deadly viruses, Nipah, Hendra, and Ebola. And so we are working, using the same system to work on a multivalent pan-coronavirus vaccine. We set up all these animal models, 3 different animal models that I told you about. So 2 mouse models and 1 hamster model, which are all ready to go to test various drugs or vaccines. And we are doing more tests of other drugs as well. I told you about this compound, a little bit about this compound that inactivates envelope viruses, and that we had really good results with influenza virus, and we're going to see if we can use that for other viruses as well, including SARS-CoV-2. And then I told you that the intranasal application of antiviral N0385 protects this mice, the humanized mice, we call them, from SARS-CoV-2 severe disease and often from death. And so I want to end here by thanking the people who are actually doing all this work. I'm just guiding them, but they are really the phenomenal people. And so the people in red here are the people who produced most of the data that I show you. So David Buchholz, a graduate student, senior grad student in the lab, Avery Monreal, a postdoc in my lab, Sharzad, a visiting scholar from Iran, and that Duncan Ethenji, who just graduated. He's a student from Kenya who is back in Kenya now, and got his PhD just this month, early this month. Brian Imbiaka is another student who has helped a lot, and Victoria Ortega a lot with along the way with these studies. So these are the main people, and the rest are other people in the lab who are not involved in this line of research, but doing other things in the lab. And obviously, I want to thank all the funding that we've gotten from Cornell and from external sources, including the NIH, DARPA, the Department of Homeland Security, and a lot of collaborators also here at Cornell and beyond, and all of the amazing IBC, BSL-3, and CARE staff that have helped us throughout the process of setting all this up. Again, most of the work that I showed you was actually done, believe it or not, in the last year during the COVID pandemic in 2020. So it's been a very difficult year, a very hard year, but we worked very hard to try to make all this work. And a lot of this, almost everything that actually everything I show you, except for that one figure at the beginning that was published in 2013, all of this is unpublished. We're working on the publications right now, on the manuscripts to send out for publication. So I'll end there, and I see if you guys have any questions. I think I'll stop the share.
Yeah.
And just to see more, or we can see, maybe I won't see the people, but at least they can see us a little more. And,
Okay.
Yeah, and
All
right.
Right.
Maybe we'll go through some questions and see if I need to share again, I'll share again my slides.
Okay, thank you so much, Hector. Yes, so far I'm seeing 4 questions in the chat. I'll start with the came in first from Peter Davies, and he later had another question. How do you produce viral particles in bulk?
Yeah, great question. So obviously in the lab, we have these cells. They're kind of factory cells, you can call them. So they're human mammalian cells. They're called HEK293T cells. And they actually transfect really well. You put DNA plasmids to express your protein of interest in the cells, and then you infect them with a virus, and there's different techniques you use, but they actually are really good producers of virus. So you can get very concentrated amounts of virus from those cells. Viral-like particles, this type of particles, are actually being used for vaccination of humans. For other viruses, there's a couple. They're all new methods. So a couple of new vaccines that just came out using this type of methodology, and they've been only used in humans for the last 2 to 3 years. But this is fairly easily scalable, basically. So you can scale it up really quickly. You can here in the lab we have our cells are in petri dishes, right? But you can actually put these cells in reactors where you're producing massive amounts of these viral particles in just liters and liters, gallons of viral particles. Here we produce them in a relatively small scale, but good enough to do all of our rodent experiments. Hope that answers that question.
Peter, did that answer your question? Okay, I thought that, oh, okay. Peter.
Peter Davis, I think.
Yeah, that was fine. Thank you. That was fine.
Okay.
Thank you.
Okay.
Thank you for your question, Peter.
The second question we have comes from Claire. And the question, I read recently that SARS-CoV-2 virus can cross the blood-brain barrier to cause its neurological effects, as well as indirectly via inflammation. How does it do this?
Yeah, endocytosis, exocytosis.
Right, yes.
Yeah. So, yes, there are a few viruses, such as the ones that I talked about today, Nipah virus, Hendra virus, Ebola virus, the coronaviruses that can actually cross the blood-brain barrier and enter the brain. How do they do that? What we know, what we understand, and not everything is understood in detail. What we understand is it does it via the olfactory nerves, okay? The olfactory neurons that go from your nose, basically, they're connected to your brain, okay? And so these viruses are able to infect those olfactory neurons and carry the virus basically through those neurons connections, right? And they'll basically enter on one side of the neurons. These neurons have very long connections extending axons to neurons in the brain, and so that's how the virus spreads from neuron to neuron. And so so through the olfactory nerves and through the nose. And so they're in the nose, for example, for SARS-CoV-2, there's goblet cells that are loaded with the receptors that the virus binds to. And so the main mode of infection of humans is through the nose, and that's why we ask everyone to wear masks, right? Because that's what the main, again, even more than touching, right? Obviously, if you touch and there's a surface with virus and then you touch your nose, you might introduce some virus to your nose, but the main thing is just by breathing it in. And those goblet cells in the nose are going to just absorb the virus, and those goblet cells are then going to infect the olfactory neurons that then will take the virus to the brain. And so that's what we understand at the time, but there's all kinds of details that we still need to figure out. Now, is it endocytosis? This virus, SARS-CoV-2 specifically, can enter cells via endocytosis, but it can also fuse at the cell membrane. So it looks like you have some good background there, knowledge on that. And so it can do it both ways. Depending on the cell type, it can directly fuse the viral membrane to the cell membrane at the plasma membrane and enter and put the RNA inside the cell, or it can actually be endocytosis first, and then the fusion can happen inside the endosome between the viral membrane and the endosomal membrane to release the RNA into the cytoplasm of the cell. Does that clarify your?
Yeah, I'm hoping I don't see.
Yeah, that's great. Thanks very much. That was
okay. Great.
Great seminar too.
Thank you for the.
Thank you. Peter Bruns has a question. Why is the virus able to replicate in bats, but it's not pathogenic in them?
Yeah, that's a question I get every single time I give this talk, and it's a really great question. The true answer is we don't know the answer to that question. It's a very interesting question. We are collaborating right now with a bat immunologist or somebody who has a colony of bats at Colorado State University, Tony Schuntz. And we have just last week found a very interesting result. We did a little study, okay? Again, this is all unpublished. We did a little study where we gave bats a high protein diet, a normal protein diet, right? And then we gave another group of bats a non-protein diet, so basically a weak diet, right? So the purpose was because our hypothesis was that when bats are not very strong so their diet is not very good, which happens a lot in the wilderness because of deforestation. We humans are going in and causing deforestation in many areas where it's the natural bats' habitats, right? And so we are getting rid of fruit trees that they feed from. And so the bats are getting closer and closer to cities. They're more and more in contact with humans. And so also our hypothesis, just from patterns that we've seen, Nipah virus, for example, that the viruses are shed more in the winter in Australia, Hendra virus, as opposed to in the summer. And so these things have led us to the hypothesis that malnourished bats, right, are going to maybe, that's known, malnourished bats, it looks like they're shedding more virus. But why? And why is that? And one question we asked, our hypothesis was that the malnourished bats would not make enough neutralizing antibodies. Okay, so we put a vaccine. We vaccinated the animals with a Nipah viral particle, like a vaccine that I told you about, and vaccinated the animals, a group with very well nourished and a group very badly nourished. And what we found, interestingly, was that exactly the opposite of what we thought was going to happen. So badly nourished bats actually produce better neutralizing antibodies than the well-nourished bats. And we are really right now, I mean, literally we got this data like 2 days ago, and we're just have this puzzle in our head. What we're thinking is that the immune system of the bat somehow is behaving differently from that of a mouse or a hamster or other animals. There's something particular about the bats. They can carry so many viruses and they don't get sick from them. And Nipah virus is a perfect example. SARS-CoV-2 seems to be another example, is looking like, where the bats can carry them, they don't get sick at all. They're shedding the virus, they're transmitters of the virus. And one question that a lot of people ask me also, with respect to them, related to that, to your question is, well, why don't we just get rid of the bats, right? And sure, that's a very, I guess, convenient human thing to say, but you got to remember the bats are super important for fertilization, and they have a lot of functions. They eat they eat insects, a lot of them, or they're important for flowering, for fertilization to make fruit. And so they're way more important. We don't see them as much because they mostly live at night, but they're actually super important in many, many areas of biology and ecology.
Thank you. Wow. Peter Davies had another question as well. A 30-year-old idea is to incorporate a vaccine into a plant food, especially for developing countries where vaccine distribution could be difficult, and perhaps incorporating them into bananas. Any possibility of this?
Yeah, I know that there's at least one group that I know of that is actually trying something like this to make vaccines from plants. And I think it's a great idea. I applaud it, and I have no expertise in plants, but I know there are, I know there's several, there must be several groups. I know there's one even here at Cornell, I believe, who is trying something like this. And so I think the plant systems are becoming better and better at doing these sort of things. One of the difficulties with that system, I'll tell you, is that these are glycoproteins. You remember I've been calling in glycoproteins, the F, G, S proteins on the surface of the virus. The reason why they're called glycoproteins is because they're full of sugars. So they have glycans. The protein stock basically has glycans coming out of that. So those glycans are actually different. The machinery in cells that makes those glycans, right, and that puts them in the right place, it's very different in plants than in animals, than in bacteria, and so you gotta make sure that the glycosylation is gonna happen somewhat naturally. And so I think it's easier to make in mammalian cells those vaccines and those proteins, just because they're going to be more natural looking. And so the immune system will make antibodies against them and protect against the real virus more effectively. And so that's one thing to keep in mind, that might be a place where I think where there might be some difficulties. But hey, engineers and plant scientists are amazing, and I hope that they can succeed in these efforts, which are really, really great.
Okay, follow
Hector,
up.
and I invite some our participants other questions. I have a question also. I was wondering, Hector, all of the work that you and others are doing here at Cornell and other universities, how does that research relate to or the pharmaceutical companies who wind up mass producing? How does that whole thing happen?
Yeah, so that's a great question, Lorraine. So here in every university, including Cornell, there's always a kind of a invention office, right? So patents and all that. A lot of times what happens is they are the liaison between the labs, the academic labs, such as mine, and the companies. And there are many different possibilities. An academic lab, the head of an academic lab can actually start a company, right? And a place like Cornell, but what happens more frequently than that is that there's partnerships where a lot of the basic research may actually, if there's an interesting company in mass producing this new vaccine or antiviral, therapeutic, they can partner with the academic lab, and if there is a patent application or something like that, they can partner to make that basically into a license. So the company, obviously, all companies want to profit. Very few companies are non-profit, right? Not for profit, but they have to work through all of that negotiation and whatever it is that they have to do, all the paperwork to be able to come to commercialization of that item. And obviously there's more involved with that, with FDA approval and all these things that companies are more experts at dealing with. We academic labs, we do what we can. We are paid our salaries from the state, and from, we get our grants from the federal government and from other agencies, private organizations. And so we are supposed to do the research, and we present, we publish, and then others are going to use that to push it further. But with SARS-CoV-2, I think we've seen an unprecedented level of connection between labs and companies to push things forward as quickly as possible for everyone's benefit.
Thank you. Peter Davies has another question. How important is the glyco? Would a protein only work?
How important is a glycoprotein? Sorry.
Yeah, Peter, you can unmute yourself and explain your question.
The question is, you're talking about glycoproteins all the time. What if you had a vaccine of only the protein? Would that work, or is the glyco part essential?
Yes, it depends on the glycoprotein. That's a great question. A lot of glycoproteins, for example, being made in bacteria, do not work very well and producing bacteria, because they don't really have anything near to the glycosylation. Sometimes no glycosylation at all happens if you try to produce the protein in bacteria. A lot of times it doesn't work, but sometimes some proteins that are just lightly glycosylated, for example, some proteins, like the HIV glycoprotein has 10 or 12 different glycans in it, and they're huge, huge things. There are some glycoproteins from viruses that have a single glycan. And so those are more likely to work without the glycosylation machinery. And so the protein itself may have, some function. The problem is folding. Proteins use the glycans to fold properly. And a lot of times, if you don't glycosylate a protein, you're gonna produce it a lot of times, but it's not gonna have the same structure. And so it's going to not produce the same antibodies also as the natural one. And so it might not be so protective. So there's a lot of challenges, but again, there's a lot of engineering being done in that area. Hope that.
Thank you. I'm not seeing any other questions in the Q&A or the chat. If anyone has any last questions, Hector, thank you so much for this. And thank you all the attendees who've come and asked questions, which helped. And as a social scientist, I must agree, you did challenge us. Put us in, helped us to get out of our comfort zone of understanding some of these issues, but I do understand them better now. So thank you so much.
Great. Great.
Any final questions or comments from our audience? Not seeing any. Again, thank you, Hector. Thank you so much.
Well, thank you for the invitation, Lorraine, and my pleasure. I love talking about what we're doing to others.
Okay, great, right. Yeah, it's really good to know that all this is going on. Okay, thank you. Thank you again. Thank you, everyone.